Hello, everyone. My name is Takoya Green. I'm a current senior in biology major here at Oglethorpe University, and today I will be talking about the effect of microplastic on ocean marine ecosystems. I chose this topic because of my marine ecology class. We've been talking about the effects of climate change on marine ecosystems, ways to combat the climate change of the marine ecosystems, as well as how the climate change of just the marine ecosystem itself impacts the world around us. And lastly, microplastics play a huge role in climate change, and especially in marine ecosystems, they have a large effect on the organisms itself. So the first thing we'll discuss today is the background information. The background information will consist of defining microplastics and relating the relevance of microplastics to marine organisms. Then we will transition into our paper. We will discuss the goal of the study design, results, and we will end with take home points to bring everything full circle. Microplastics are small. What are microplastics? Small pieces of plastic smaller than 5 milliliters in length that are harmful to the ocean and marine ecosystems. Microplastics are depicted on the right. This is a hand, someone placed microplastics within the sand into their hand. And although these microplastics don't seem as large, they're very harmful to marine organisms because their marine organisms are unable to fully digest these microplastics. And these microplastics contain toxins and contaminants. Microplastics relevance to marine organisms. Why should we care about microplastics in marine ecosystems? The first reason would be the food web transfer. The food web starts with a primary production and goes all the way up to consumers, 3 to 4 levels of consumers from starting off. And there is the food web is a inverted triangle. So we start off broad with primary producers such as plankton and go all the way to the top predators, which can include sharks, whales, dolphins. And if microplastics are, if zooplankton are ingesting microplastics, and these zooplankton are being preyed upon from their predators, the microplastics are just further aggregating up the food web and being ingested by each organism that is ingesting the previous organism, which creates a cycle. The next reason is for physiological effects. Microplastics can cause a reduced growth rate. They can alter feeding mechanisms as well as cause reproduction failure. Due to these microplastics not being fully digested, as well as the toxins and contaminants released that can alter body functions within the homeostasis of the organism itself. Lastly, would be the physical injury. Microplastics can cause abrasion cuts within the organism, which can alter their feeding mechanism. And if they're not able to maintain nutrients, it decreases their survival rate, as well as other abrasions outside of the body, such as cuts. And if they're not fully able to recover, this decreases the survival rate as well. The infographic on the right depicts the microplastics in oceans. So, commonly found plastics in the ocean consist of cigarette butts, bottle caps, straws, cups and plates, single-use bags, food wrappers, and beverage bottles. And there are various methods of how these plastics are transferred to the ocean. And the first would be through weather, such as rain and wind, littering, which is something done by humans, streams and storm damages, dam drains, streams and storm drains, which are often lead to the open body of water, such as oceans. Then we have items such as boats and nets, especially in things such as large commercial fishing. We have smaller microplastics that are being ingested not only by fish and smaller species such as zooplankton, but as well as sea turtles and higher up organisms, as well as larger plastics, which are known as macroplastics, which are the entanglement of organisms being entangled in these plastics. The goal of the study. The purpose of the study was to examine the distribution of microplastics and quantify this distribution amongst different species. The experimental question was, how does the food transfer web of microplastics affect higher trophic level species? Lastly, the hypothesis was microplastics are aggregated up the marine food chain through different trophic levels. The study took place in Zhanghan Fishing Ground, which is located in East China Sea. This is one of the largest fishing grounds in China, and the fishing, the actual collection of species took place from two of the ports, the A1 and A2, across a 2-day span. The species were collected using a bottom trial method. A bottom trial method is a large net being dragged across the ocean floor and just collects anything in its way. This can include small species, large species, plastics, pollution, anything like that. And there in this study, a 32-meter net was used to collect the data over two spans. 19 of species were collected during this collection. 136 of the individuals 136 of the individuals collected were crustaceans such as crabs and krills, and 196 individuals were fish species that were collected. Once the species were all collected, a basis sample collection was done. The sample collection included gender, body mass, full index body mass, health, the condition of the species. The reproduction tracts were also examined. And once the baseline sample collection was collected, the species were then, the species gills and GI tract was then sampled for the digestion aspect of the study. So the study was to examine microplastics ingested in species such as fish and crustaceans. And so during this, researchers took tissue samples and probed them and placed them in various solutions and let them sit for 48 hours. And after the 48 hours were completed, they were then examined through microscopes as well as photos to do a sample collection of the microplastics. Figure 1. Figure 1, the average abundance of microplastics. On the y-axis, this consists of groups based on what the actual species was, as well as they are divided into groups based on how many individuals of that species was collected. So if you were in group 2 means that 29 or less individuals of a certain species was collected during the bottom trial method. Group 1 indicates 30 or more individuals were collected during the bottom trial method. And on the x-axis, we have microplastics per gill item, microplastic item per gill, and then we have microplastic item per GI tract. And in group 2 of the crustaceans, we see an abundance of microplastics in 2 species, which are about the same, but one is microplastic is abundant in the gills, while the other species microplastics are abundant in the GI tract. In group one of the crustaceans, we see an abundance in both microplastics in the GI tract and the gill tract in two species. And the abundance of microplastics seem to be more abundant in the GI tract for this species, and the microplastics abundance seem to be about the same in both species, but in this species, the abundance of microplastics are lower. And in group two fish species, we see an abundance in microplastics, of a few species. So we have both abundance of microplastics in the gill and the GI tract, which are about the same. They're at a 0.5 average. Then we see an abundance in the GI tract for this species, and then an abundance in the gill tract, the gill of the species. Lastly, group one, fish species, we see an abundance of microplastics in both the gill and GI tract. And this species, the most abundant microplastics are prevalent in the gill, while although the microplastics in the GI tract are abundant, the gills have more microplastics as seen in this area. And the averages were based on the microplastics were average, and then the mean average was taken. And that is how we're assessing if it's a 2 or if it's a 0.5. The most abundant items in the GI tract of microplastic items in the GI tract is in group 2 fish species with a approximately 1.3 average within the GI tract, while the most abundant microplastic in the gill is approximately 2, has a 2 average mean in the group 1 fish species. And this would make sense, this would correlate because there are 30 or more individuals that were collected in this sample in comparison to this. Figure 1 is just the microplastics data collected and averaged based on the amount of microplastics found in the sample collection of the tissues. Figure 2. Figure 2 is the chemical composition of microplastics identified in the gills and the GI tract. The left figure represents the gills, the right figure represents the GI tract. And in both figures, we see that PET polyester is the most abundant microplastic prevalent in both the GI tract and gills. And the reason it would be is because PET and polyesters are used in a lot of our everyday life. For example, water bottles, cleaning supplies, laundry supplies, anything like that. Many of those items use PET and polyester. And we see that it's at a 36%, which is not quite half in the gills, but at a 58% in the GI tract, which is the most abundant and is more than half of the other microplastics that were surveyed throughout this experiment. Next, we will talk about the distribution of microplastic counts versus size in fish individuals. So the y-axis represents microplastic counts from 0 to 20. And then on the x-axis, we have a size distribution based on micrometers. And then we have the cumulative frequency of the two. So as we see in the line representation on the graph, microplastics in the GI tract of fish species and microplastics in the gill of the fish species are about the same. There is they have a similar correlation. We do see that the microplastic in the gill species is a little bit higher than the GI tract, but eventually once we reach the maximal size, they both meet at the frequency of 1%. We also see that the bar representation of the graphs show that microplastics are abundant in the smaller size distribution of the species. And then we do see microplastics in gills. So the most abundant is the microplastic in gills, but there are higher numbers of microplastics in the GI tract as well. And we see this all the way out until we get to our largest size distribution. And then that's when we see um, microplastics to be more abundant in the GI tract out here, as opposed to the microplastics in the gill of the fish species. Next is figure 4, which is the relationship between trophic level and microplastics abundance in crustaceans. So the last figure depicted the abundance of microplastics based on size distribution, which talked about how microplastics are aggregated up the food web, and it was depicted. Now this one is this figure is correlating the trophic level to microabundance, but instead of in fish, in crustaceans. And as we can see, the higher the trophic level is, the more abundant the microplastics are. So the y-axis shows the trophic level, the x-axis shows the different species that were collected in the sample data, and then this axis shows the abundance the microplastic abundance per individual. So as I said before, we see a high correlation, a strong correlation between trophic level and abundance. So the higher the trophic level is, the more abundant the microplastics are. And then as we continue to go down, we see an inverse. So the mid level of the trophic level, the abundance of microplastics are lower. And then as we continue to get further down, the abundance of microplastics increase in comparison to trophic level. And then they both meet, meet kind of like at a equilibrium of trophic level versus abundance. And this is what we expect to see, because we expect the trophic level, as trophic levels increase, the abundance of microplastics increase. But in the mid section of the trophic level, in comparison to the abundance of microplastics, this is not surprising because we see an abundance of various species in these trophic levels. So the case may be that some of these crustaceans are not preying upon some of the lower trophic level species that are consuming these microplastics due to the variety of species that can have a predator-prey relationship within the inverted triangle of the food web. Lastly, take-home points. So the biggest take-home point is that plastic pollution has an impact on our marine ecosystem. Not only is there one particular species or one particular trophic level being impacted by the debris of plastic, all of them. We say transfer of microplastics from our basic primary producers all the way up to our top predator. Next is that the study showed consistent data of higher correlation between microplastic abundance as trophic levels increased. So this is like depicted in the last two figures. As the trophic level increased, the abundance of microplastic increased, meaning that these larger predators are consuming more of their prey and ingesting more microplastics. Lastly would be that zooplankton, which are primary producers, are ingesting microplastic, and this is the starting pathway for microplastics to be ingested from in the food web. So if they're ingesting microplastics and and then they're being preyed upon, this just microplastic is going up the food web and creating a cycle of microplastics that and is continuous. Lastly, the most commonly found microplastic is the PET polyester. As stated before, these plastics are found in many of our everyday life. And one way to help combat these plastics from being polluted in areas of our ocean is cutting back on our consumption of these PET and polyester items, such as water bottles. This figure to the right sums up the take home points. And it just shows the microplastic convergence of the food web chain. So on the first trophic level represents the plankton, which are primary producers. So, and this can include corals, this can include any of those organisms. And then we see the second level go up. And these dotted lines represent the direct predator prey relationship between these primary producers and the next consumers of the food web. And this continues to go on and on. And then we see that these lines represent the indirect predator prey relationship, but there is still a relationship, although these predators are not primarily consuming the lower prey, but they are being impacted. And this is how microplastics are being passed up upon our food web. And not only does microplastics in marine ecosystems affect the organisms in the marine ecosystems, they affect humans as well. Thank you for watching my talk. I hope you enjoyed.